The last topic of this lecture is membrane proteins, and kind of zooming in a little bit and seeing how they function. In particular, I want to focus in on their ability to help with transport. So I want to think about transporting molecules across membranes. And so we're going to think of a few different scenarios here, one of which would be if we're talking about small, uncharged molecules. These would be things like oxygen, carbon dioxide, water, right? So rather small things that don't have charges. And these things can diffuse right through the biological membrane without any assistance, really. And so it might look something like this. So let's think about the membrane as being the squiggly line. And we've got, say, outside the cell, and we've got inside the cell, like that. And there's some molecule A. And because this molecule in this example is really small, it can just pass right on through. And so we call that non-mediated transport. Right? Non-mediated because there's no mediator needed. This molecule can go right through the membrane. Now, on the other hand, if the molecule that's going through the membrane is large or charged, so things, for example, maybe like sodium has a charge, or maybe something pretty big like glucose. Now, these are too big. Glucose is too big to just squeeze its way through the membrane, and sodium, because it's charged, is going to recruit a bunch of water molecules to hang out with it, and so it effectively becomes a big molecule, or big ion. And so it's not going to be able to go right through the membrane. It's going to require a transport protein, and so we would call this a mediated process. That protein is going to mediate the transport. So that might look something like this, where we've got the membrane again. Here's the outside and the inside. And now, rather than just passing right through the membrane, we're going to need a membrane protein right here. So you can indicate that as a protein. And as long as, there's a lot of whatever that A molecule is, sodium or glucose or whatever, and not a ton on the inside, then it'll kind of be easy and spontaneous for that molecule to go in, using that protein as a door, essentially. And so in this case, I've just shown these molecules going down their concentration gradient, right? So there's a lot of the A molecule, glucose or sodium or whatever, on the outside, and there's not as much on the inside, and so it's favorable. Think of like entropy. It's favorable for this to want to equalize out a little bit. So it's okay for A to enter the cell. That's energetically okay. We call this a passive process because it doesn't require any energy or anything can kind of happen with just having this protein present. It doesn't require pumping any energy into the system to convince that molecule to go the other way. Now, in contrast to that, we might have the reverse scenario where, again, we've got outside the cell, inside the cell, and still the molecule is going to need a protein to get through, but it's the opposite scenario. Maybe now there's a little bit of sodium or whatever on the outside and a whole lot of the A molecule on the inside, and we need then to get this A molecule in, but that's not favorable. The concentration is low outside and high inside, and that, from an entropy disorder standpoint, is not favorable for the molecule to move that way. This is, as we would say, is moving against the concentration gradient. And the only way that can happen is if we put energy into the system somehow or another, just like a pump, right? We can't get water to go uphill unless you pump it. Pumps require energy. But if you are going downhill, down the concentration gradient, water falls over a dam spontaneously because it's favorable to do that. So it's not a passive process in this case, it's actually an active process, and so we would call it active transport is what that process is known as.